Now we go to the next speaker, if he's available, Mahendra Dewal. Uh, I saw him actually. He was here.
Yes, I'm here, sir.
Yeah,
Yes,
thank you.
I'm here.
Founding member, Expansion Technologies, Boston, USA. MC, please introduce Dr. Mahendra B. Dewal.
Dr. Mahendra Dewal. Dr. Mahendra Dewal is a creative and self-motivated scientist with outstanding 7 years of academic and 5 years of industrial research experience in multiple disciplines, including expansion microscopy, biochemistry, molecular and cell biology, etc. Dr. Mahendra Dewal is efficiently managed team-oriented project stand, co-writer of founded research proposals from Mizutani Research Foundation of Glycoscience. Dr. Mahendra Dewal is a team player with interdisciplinary scientific experience and provides a unique ability to interact and effectively collaborate with scientists of various backgrounds and work on multidisciplinary projects. Dr. Mahendra Dewal has multidisciplinary professional skills and is also expert in molecular biology, gene cleaning, cell biology, and in organic chemistry. Now let us welcome Dr. Mahendra Dewal to deliver his note. Thank you.
Thank you, Divya.
Thank you. Thank you very much, Divya, for your kind words about my introduction. Good morning and good evening, everyone. First of all, I would like to express my immense gratitude to the organizers, Dr. Hari Ippanapalli from Lead India Foundation and entire his team members. And also Dr. Ram Maruri from Induct and Library Foundation and entire his team members for giving this opportunity today to speak at this global webinar, great event, and conducted in the memory of our beloved great scientist, great leader, 11th President of India, Dr. Abdul Kalam Garu. Moreover, Dr. Abdul Kalam was an inspiration to many, many, many young minds from many decades. I'm none other than one among those students who inspired by reading his books, watching his speeches, and learned a lot. And I became a scientist inspired by him. So I also thank everyone who is sharing. It's a great opportunity. It gives me an immense pleasure for me to share this virtual stage with many, many great people on this call today. Thank you. Thank you so much for being here. So as a scientist, I'm going to share my thoughts in healthcare, biotechnology, the opportunities and challenges in biotechnology. Mostly, I will be focusing on development of next generation biotherapeutics. The next 2 decades are the opportunities going to be open, many, many opportunities for the various different disciplines, students, young scientists. The many opportunities are going to open in next generation biotherapeutics. If you look at this biotherapeutics area in the past 3.5 decades, the focus of drug discovery and development has gradually shifted from small, well-defined chemical molecules to a large, complex biologics. And because there are so many unmet medical needs, because, for example, lots of genetic disorders, chronic diseases were not cured by these small chemically well-defined molecules, but biologics are playing an important role in the past 3 decades. And they are also playing very prominent role in combating diseases like cancer, diabetes, and many autoimmune diseases as well. In the next generation biotherapeutics that I'm going to talk are going to influence mostly in genetic disorders, curing the genetic disorders, in regenerative therapies, and personalized medicine as well. So in this topic, I'm going to talk about various different therapeutics, such as engineering the proteins and antibodies, cellular therapies, gene therapies, gene editing technologies, and RNA therapeutics, as well as vaccine development technologies. If I still have a time, 1 minute, I'll quickly introduce the organ transplantation, which is called xenotransplantation, transplanting organs from different species to different animals to humans, and the opportunities that are there in these different fields. So antibodies. In the past 3 decades, monoclonal antibodies were developed and utilized as a drug candidates for various different diseases, including rheumatoid arthritis, psoriasis, Crohn's disease. One best example is a drug called Humira. It's also called adalimumab, which was developed by Aviv Therapeutics, and it treats the diseases that I just mentioned, like rheumatoid arthritis. It generated global sales about $20 billion in 1 year, that is 2018. So, which is going to be double and triple in the next coming decades. But this is already developed. But next coming generations, what kind of technologies are in the antibody engineering are utilizing? So now mostly the scientists are trying to develop antibody conjugates so that they can selectively target the cancer cell or tumor cell or disease site. So you conjugate the drug molecule to antibody and send it to that particular cancer so that the cancer cells will be removed easily. That's one technology. There is a lot of opportunities are growing. The second thing is bi or multi-specific antibodies. Generate one antibody but treat multiple diseases. So it is specific to 2 to 3 different targets. That's another area. And also take a fragment of antibody. Antibody is a complex biological large molecule. But if you engineer a small part of the antibody, which is called nanobodies, there also huge amount of opportunities are there for the next generation students. And the second area is cellular therapies. The cell therapy is nothing but transplanting the cells into the patient's body to cure the disease. These cells may come from the patient itself or from a donor. So what we do is, you take the cells from the patient or a donor, and then you engineer them, put them back into the body, the patient's body, so it will cure the disease. Oncology, musculoskeletal disorders, autoimmune disorders are cured by these cell therapy already being approved. There are 2 drugs, Chimera and Yescarta, were already approved, which is also called CAR T cell therapy, to treat acute lymphoblastic leukemia and as well as beta cell lymphoma. It is estimated to acquire about $10 to $15 billion of market in the next 5 years of these 2 cell therapy drugs. Next method is gene therapy. Gene therapy is using the genes to prevent the disease. There are 2 methods in here: adding the new genes, delivering the new genes into the body, or the repairing the genes that are already malfunctioning. In these 2 methods, first I'm going to talk about the adding the new genes. So mostly viral vectors are being utilized. The engineered viruses, make them unharmful and then use them to deliver this particular gene that is malfunctioning in the body, that is causing the disease, to fix the gene and cure the disease. And there's a recently approved Zolgensma, which is a new drug that approved to treatment for the pediatric patients with spinal muscular atrophy. Most prominently, adenoviruses or adenoviral associated viruses are being utilized to deliver these genes into the body. And there is already about 900 other potential therapeutics, and gene therapeutics are in clinical trials, ready to approved by FDA in the United States. Next one is gene editing. The CRISPR-Cas9 is an efficient and versatile gene editing technology. This was awarded with a Nobel Prize in Chemistry last week for the 2020. This is a unique technology that can repair the genes, malfunctioning genes. In this method, in this approach, there are 2 methods. One is fixing the gene ex vivo or fixing the gene in vivo. Ex vivo means outside the body. So you take the cells out of the patient and you induce these editing components such as a Cas9 enzyme and an RNA into the cell and repair the gene, put the cells back into the patient's body so that the disease will be cured. The second thing is deliver this whole component using the viral vectors or lipid nanoparticles into the patient's body and fix the genes inside the body to cure the disease. These are the 2 methods, but currently mostly ex vivo gene editing is utilized. There are a lot of therapeutics are being under development in clinical trials for hemoglobinopathies such as sickle cell disease and diabetes, and also in immuno-oncology. Next one is mRNA therapeutics. mRNA therapeutics, RNA, generally protein is coming from the genes, right? DNA transcribes into RNA, RNA translates into protein. That's which functions, its functions in the body. So RNAs can be utilized as a drug molecules. There is a lots of opportunities, many like small interfering RNAs, ASOs, microRNAs, regular RNAs are being utilized. There are lots of drugs are being developed to cure the diseases using the RNAs as well. Lots of opportunities also are there. And I'm going to take not more than 2 minutes. So we are in the middle of the pandemic, and there is novel vaccine development technologies also. I'm going to give an introduction in 1 more minute. So we are in the middle of the pandemic of COVID-19. We desperately need a vaccine. About 3 decades ago, there are only 2 methods to develop the vaccines, such as using the live attenuated viruses or using the inactivated viruses. But in the 3 decades, various different methods are developed, such as using the viral vectors. They can be replicable or non-replicable, creating the vaccines, and using the nucleic acids to create a vaccine, such as DNA or RNA or protein-based methods. In the case of COVID-19, coronavirus spike protein is a best target to generate the vaccine. Many companies in public and private partnership, there are approximately 193 total potential candidates are under development, and 42 of them are already in the clinical trials. At least 5 to 10 of them are in the late stage clinical trials, which may be available anytime from now to protect from the COVID-19 infections. Like I said, there are so many opportunities are there in these various different biotherapeutics areas, but there are also challenges. For example, the quality control procedures are very stringent, and these are complex biological molecules, not easy to synthesize them. The manufacturing viral vectors is not so easy, very time consuming. Generating the robust methods are also very difficult, and the pricing is very high at the moment, but all these challenges can overcome with the time. As I mentioned, the market is growing billions and billions of dollars. Next 2 decades are going to be the market for these biotherapeutics. Having said that, thank you so much. If you have any additional questions, feel free to reach out to me on the social media or LinkedIn or anywhere else. Thank you so much for the opportunity.
So thank you very much, Mahinder, for joining us and sharing your wonderful insights. And I'm very proud to see you. You
My
are
pleasure,
the front
sir.
runner on the genetic side, and we will talk more about it as we progress. So
My
thank
pleasure,
you again.
sir.
Be safe.
Thank you, sir.
Yeah, thank you, Mahinderji, once again. Now